My name is Nadeem Samaan, and I'll be presenting our grant proposal along with my team members, Edgar Cortes and George Demestihas. And we are investigating the rate of cancerous cell division when GFP protein drug is administered at different dosages to melanoma-induced C. elegans. Melanoma is a serious form of skin cancer. It develops in the cells that produce melanin, which is the pigment that gives our skin its color. We currently don't know the exact cause of melanin, but we do know that high exposure to ultraviolet radiation from the sun or from other sources will increase the risk of developing melanoma. Looking at the drugs that treatments we have today for melanoma, we have more room for a better and more effective drug. The plant Greenglow funcadelicus is a plant native to the Amazon rainforest. It is used by the Amazonian natives to heal skin wounds, and it's believed that the green fluorescent in the plant is responsible for its ability to heal the skin. And that green fluorescent is expressed by the green fluorescent protein, which is found in the plant. It's determined that this protein can potentially be used in a new drug to treat melanoma. In order to test our drug, we need to use a very effective model organism. So we chose C. elegans because they have the organs that are very similar in form and function and genetics to those of the humans, and they have relatively short life cycle, which makes them a great model organism. So in order to use the GFP gene or GFP protein for the drug, we need to produce the GFP gene in bulk. In order to do this, we used a molecular cloning approach. So in the beginning, we extracted the DNA, the GFP sequence from the plant cell using restriction enzymes. We used the same restriction enzymes to linearize the vector to make sure that, and the reason we do so is to ensure that they're compatible and they ligate in the right direction. So we prepare the cells using calcium chloride to transform the plasmid into the E. coli cells, the P-bad plasmid. We grow the E. coli cells on plates, and whichever plate shows the green fluorescent, we will take those colonies and we will let them grow in a liquid medium to allow for more growth. And we will isolate the plasmid using mini-prep, and we will use PCR and gel electrophoresis to confirm that the insert of the GFP was successfully cloned. And after that, we will purify the GFP protein so we can use it for the drug. This is an image of the P-glow plasmid. This is our GFP gene. The restriction enzymes we used are NHEI, which cuts at 1345 base pair. And the second restriction enzyme is the EcoRI, which cuts at 2063 base pair. The restriction enzymes function at 37 degrees Celsius. The primers we designed specifically for the PCR are the GFP forward primer, which has a 47% GC content, and the GFP reverse primer, which has 58% GC content. So when we did our preliminary experiment, we had two main samples. The first sample has the E. coli cells as well as the ligation product, which is a ligase, which is uh, the, the plasmid. Um, so panel A has the E. coli, on the plates, the E. coli and the plasmid. However, in panel B, this is our negative control. So these will only have the E. coli cells without the plasmid. All plates have luria broth in them to provide the minimum nutrients needed to grow the cells. Um, only plate 2, only the second plate has the all select screening reagents used, which are the ampicillin and the arabinose. The first plate only has the ampicillin but no arabinose. The third plate has ampicillin and no arabinose, and the fourth plate has it only has the luria broth. It doesn't have any screening reagents. So as expected, it would not show any bacterial colonies. Um, we do expect the second plate, number 2, to show the green fluorescent because it has the E. coli and it has the plasmid and it has both screening reagents. So from here we choose 4 bacterial colonies to extract the plasmid via mini prep, and we run in the PCR and gel electrophoresis. So after the PSR is performed, the PCR products are run on gel electrophoresis, and this is our data for the gel. Starting from the left, we have the ladder. After that, we have our positive control, which has the GFP gene. The negative control, which only has the PBAD plasmid. And then 1, 2, 3, 4, those are our colonies that we chose from that growth plate, from the second growth plate, which has the green fluorescent showing. And this is our experimental control, which has all the reagents except for the DNA. So looking at this data, we can see that almost all of our colonies have this band at approximately 750 base pair, which is the same as our positive control. And we know that the positive control is the GFP gene, so we can confirm that the banding length, the base pairs, and the pattern is the same as the GFP gene, so we can confirm that in fact we did successfully clone or insert our GFP gene. So after that, we can proceed to purify our protein, the GFP protein, and we will use a protein affinity chromatography approach. The way this works is we use affinity resin, which has ligand attached to it. This will have a high affinity to our target protein, indicated here. So this is our GFP. This is our target protein. All the other things here are the other proteins and the other, basically any cellular debris. And we only want our target protein. So when the ligand attaches, when the ligand is bind to the target protein, it will make sure it's left behind when we perform our washing phase. So after we perform our washing phase, all the other proteins will go away, and we will be left with just our target protein, which is the GFP.
So the aim of our experiment is to investigate dose dependency of GFP protein on induced melanoma C. elegans. As you can see in the cartoon, the pink bubblegum shaped organism is supposed to represent C. elegans, and it is eating GFP, which is our basis of our experiment. So for our timeline, it consists of running the experiment for 40 days. So on day 0, we're gonna receive our C. elegans model organism and confirm that the C. elegans are induced with melanoma. On day 1, we are gonna place the C. elegans on agar petri dishes containing E. coli as the food source. And next, day 2 to 4, we're gonna allow the drug to perform its mechanism of action. On day 5, we're gonna observe and conclude if different dosages of GFP protein have effect on melanoma after 4 days of the initial exposure. On day 6, we're gonna continue to make conclusions about the reversal effect of melanoma of GFP protein at different dosages. And we're gonna record if any model organism died or any negative side effects developed, for instance, like lesions or any other type of negative side effect. The next is going to be the next 7 to 40 days is we're going to just review our data and decide whether the drug should continue to human trials. So there are multiple ways to determine the efficacy of the drug dosage. The first is macroscopy, and this is used to used on live C. elegans to determine the morphology and locomotion of the C. elegans. The next piece of equipment is called an automated microscopy, and this is used to determine immunobilized fluorescence and morphology. The next piece of equipment is called a microplate reader, and this is used for live C. elegans and immunobilized fluorescence. And this piece of equipment we are going to use for our experiment specifically. So the specific type of microplate reader we are going to use is called an array scan VTI, produced by Thermo Fisher. And so it's just specifically described as a robotic fluorescent-based imaging system. And so how it functions is it takes several pictures of the petri dish of our sample, and we'll be able to see the fluorescence of it. And next, another piece of equipment that we will be using is a spot detector bioapplication, and this will be used to see the fluorescence specifically. For instance, when we use this will be coupled with the scan array VTI, and when it's coupled, we'll be able to quantify cell division, melanoma, and cell death. So in order to see fluorescence, we have to have markers. So for our biological markers, we're going to use histone H3, which is our cell division marker, S100B, which is our melanoma marker, and SYTO12, which is our cell death marker. So for our experiment, we're going to have 4 different groups of C. elegans. The first group is going to have no dosage, 0 micromoles of GFP protein. This next group is going to have low dosage, which consists of 10 micromoles of GFP protein. Then the third group is going to be exposed to medium dosage of GFP protein, which consists of 25 micromoles of GFP protein. The next group and final group is going to be exposed to a high dosage of GFP protein, which consists of 100 micromoles. Each concentration of GFP protein will be spread across the agar plates on specifically the lawn. And so as you see from the image to the right, this will be the fluorescence and the colors that it will emit. It will not look like a drawing, but you'll get the idea of the fluorescence of the color. So this is mock data. So this is what our C. elegans would look like on the petri dish with its E. coli and the dosage. Obviously, it's not going to be with the fluorescence yet, but the first group is going to be called no dosage. The second group, which is a B, is going to be small dosage. The next group is going to be medium dosage, and the last group is going to be a high dosage. So this is what our possible data would look like when we utilize the markers. This image specifically was obtained by O'Reilly and his team, and they used a array scan VT ion spot detector bioapplication to investigate misfolded AT proteins in the intestines of C. elegans. In this instance, they didn't use our biological markers. So in our experiment, we're going to use the S100B, which is the melanoma marker, which will emit a blue fluorescence, a histone H3 cell division marker, which will emit red fluorescence, and then we'll use SYT012, which is the cell death marker, which will emit a green fluorescence. So if we were to apply our markers to their experiment data, we would say that in figure F, the C. elegans have a large amounts of green fluorescence and therefore cell death. And then images B and E have a moderate amount of red fluorescence, which is cell division. So we would say there is a moderate amount of cell division in the C. elegans. So for our possible outcomes, we were analyzing how different dosages of GFP protein affected the amount of different fluorescence emitted in the C. elegans model. So in this graph, we made mock data of how blue fluorescence varied depending on the dosage the C. elegans was administered. So as the dosage increases of GFP protein, the amount of blue fluorescence decreased. Therefore, we can say that higher dosage, as the dosage increased, the amount of melanoma in C. elegans reduced. This is significant because it shows that higher dosages of GFP protein can reverse the effects or treat the effects of melanoma. And next, we made a graph depicting how green fluorescence would be affected by GFP protein and the variation of GFP protein applied to C. elegans. For instance, as you see in the graph, as we increase the amount of GFP protein, the amount of green fluorescence in the C. elegans decreased. Therefore, we can say that as you increase GFP protein content, you decrease the amount of the amount of cell death. This is significant because the GFP protein is reducing the amount of melanoma, but it's also decreasing the amount of cell death, which means that not a lot amount of healthy cells are being killed, and the cancer cells are being killed. So the next graph is we made mock data of how red fluorescence would be affected by by GFP protein concentration. So as you see, as you increase the concentration, the dosage by micromoles of GFP protein, the amount of red fluorescence emitted by the C. elegans did not change at all. Therefore, we can say that GFP protein does not have an effect on cell division. This is significant because if melanoma continues to be present in the C. elegans, it's still able to undergo cell division and replicate, which will continue the melanoma. Therefore, our overall findings suggest that at a higher that GFP protein functions at higher dosages. It is more effective in treating melanoma at higher dosages because it decreases the amount of cell death. It decreases the amount of melanoma cells, but it does not have an effect on cell division. However, if GFP protein is continued to be administered at high dosages, melanoma cells can be killed completely, and therefore cell division won't be a factor accounted for because there would be no melanoma cells to divide and replicate. Therefore, no melanoma.
Hi, my name is George Demestihas, and today I'm going to be talking to you guys about the second possible set of outcomes and conclusions for our grant proposal. So starting off with the no-dose group, we see a high level of blue, green, and red fluorescence, with blue standing for the amount of melanoma, green standing for the amount of cell death, and red representing the amount of cell division that is occurring. So the high amounts of the blue, green, and red fluorescent markers in the no-dose group are indicative that melanoma is in fact acting freely, dividing freely, and causing cell death freely. Now, when we move on to the small dose group, we see how, although the fluorescence levels are high, they're a bit lower than the no-dose group. This makes a lot of sense because since we administered a small amount of GFP, there will be a small decrease in melanoma's activity, signifying to us that the GFP is in fact working in combating melanoma. Now, when we go to the medium dosage group, we see how there's low amounts of the blue and red fluorescence, and there's a moderate amount of the green fluorescence. This is basically supporting the results that we receive from the small dosage group in showing us that as we increase the dosage of GFP, melanoma will be more effectively combated and stopped. And this is marked by the lower amount of cell death, the lower amount of cell division, and the lower abundance of melanoma. Now, as far as the high dose group goes, we see a low level of both the blue and the red fluorescence, indicating that melanoma is in less abundance and cell division is occurring less rapidly. However, we see a high level of green fluorescence, indicating to us that although melanoma is being combated, GFP in the high dosage level is actually killing the host cells as well as the melanoma cells. Now, for the basic trends observed for melanoma, or the blue fluorescence marker, as the dosage of GFP was increased, we saw that blue fluorescence, or the abundance of melanoma, also decreased. This is really good because it's telling us that as GFP increases, melanoma will be more effectively combated and found in less abundance. Now, as far as the green fluorescence, or the marker for cell death, when we examined it, we saw that as dosage was increasing, as GFP dosage was increasing, the amount of green fluorescence also decreased. However, when we administered the high dosage of 100 micromoles, we found that the GFP actually caused cell death, not only in the melanoma cells, but also in the host cells. Now, as far as cell division goes, we observed the same trend as we saw in the melanoma trend. So basically, as the dosage of GFP was increasing, the amount of red fluorescence decreased. This is basically telling us that the more GFP that was administered, the less cell division would occur from the melanoma, thus indicating that the GFP was effective in combating melanoma. Now, as far as our experiment goes, we built a simple flowchart to illustrate the brief but effective multi-step process we took to test whether or not the GFP was effective in reducing skin cancer in melanoma-induced C. elegans models. So first we had to receive the C. elegans models induced with melanoma. And once we received those models, they were separated out into 4 different groups: the no dose, the small dose, the medium dose, and the high dose groups. They were placed on agar lawns with varying amounts of GFP according to their group and observed for 6 days. After the 6 days, which we deemed enough time for the GFP to take effect and melanoma to respond accordingly to it, we made conclusions about whether or not cell division was abundant, whether or not melanoma was abundant, whether or not cell death was abundant. If we found that these things were not abundant and that the GFP was in fact combating the melanoma, then we can begin the next step towards clinical trials. However, if we find that there's a problem with the experiment, we're not getting the results we want, then we'll adjust accordingly. Now, speaking of such issues, one pitfall of this experiment could be our C. elegans model organism for one reason, and that is because it has a highly selective impermeable cuticle that will not allow anything through unless it's water soluble. So essentially, certain molecules such as proteins that aren't very water soluble may not be able to be properly ingested and metabolized by the C. elegans, therefore affecting our results. Since if less GFP is getting into the C. elegans, that means that less GFP will be acting on the melanoma, and our results will be affected by showing that GFP is less effective against melanoma than we would like it to be. Now, another pitfall is that the E. coli bacteria, the live bacteria that we're using on our agar lawns to feed the C. elegans specimens and keep them alive, could potentially metabolize the GFP or degrade it. This is because it's alive. Now, if this occurs, we could potentially use heat-killed E. coli, and the reason that we would do this is because they have less metabolism of GFP because they're heat-killed, and as a result, more GFP would become available for the C. elegans to uptake, thus giving us more accurate, untainted results. Now, there are several ways we could improve this experiment. One of those ways being that we could use the heat-killed E. coli if we find that the uptake isn't as efficient as we would like it to be. Additionally, we can increase the concentrations that we use and administer simply because if there's more GFP available, then it'll increase the chance of GFP being utilized by the C. elegans model organisms and increase the chances that the GFP will combat skin cancer. Now, if we find that these methods don't work, we do have an alternative method to administer the GFP protein, and that's through microinjections. So basically, we would use a micropipette and a needle to apply the GFP directly to the areas of the C. elegans models affected by melanoma. How we would do this is we would freeze the C. elegans samples so that they're not squirming around moving so much, so that we can accurately place specific amounts of GFP on melanoma affected areas, and we could then observe for the 6-day period and continue on with the experiment as we normally would. The only difference would be that we would inject the melanoma instead of let the C. elegans uptake it passively in the agar plates. Now, to conclude this, we found that more GFP equals higher uptake, which would equal more effectiveness. So essentially, if compound concentrations are increased, the drug is more effective since more of it is available. Likewise, if we used, we found that likewise, we realized that heat-killed E. coli improves C. elegans drug uptake. So if we do find that in our experiment, the C. elegans aren't properly uptaking, there's a variety of things that we can do to improve our results, such as using the heat-killed E. coli or using the microinjection method. Now, the reason that this study is plausible is because green fluorescent protein has been previously found to treat an abundance of skin ailments. It's been widely used and applied topically to the skin directly, and it's been found to reduce symptoms as well as treat the disease. So we believe that if GFP can be used for a variety of skin ailments, then why not skin cancer? We think that with sound scientific and experimental methods and equipment, that we could get to the bottom of GFP and figure out whether or not this could be used as a potential skin cancer treatment candidate. We believe that this is actually a relevant study and that it's extremely plausible, first of all, because there's no drug that passively treats melanoma. Furthermore, there's no sound remedy for melanoma in that, like, treatments may occur and treatments may take place. However, that doesn't mean that they won't, the melanoma that's being treated for won't return, and that doesn't mean that it's completely cured. So the reason that this study is extremely important is because it could revolutionize melanoma treatment. If it is found that the GFP protein is effective in combating melanoma, it would be the next step in creating a life-changing way of treating melanoma skin cancer in a less invasive and more effective way that will improve patient survival rates as well as improve patient satisfaction and reduce cost. It could be a widely available revolutionary medicinal treatment for melanoma. Furthermore, why not learn about green fluorescent protein? It's been heralded as a great way to treat skin ailments. So we believe that this study is a good first step in understanding better green fluorescent protein and all of its potential. It's an extremely worthwhile research project to us because we believe it has huge potential. If it's effective in treating melanoma, could it treat other skin cancers? Could it treat other cancers in general? Could it treat other illnesses of the skin that we haven't yet thought of using GFP to treat?